So we have arrived at the last process in our central dogma, translation, where we're going to move from mRNA to a polypeptide. And here we're going to be switching languages. We're going to be translating languages, going from the language of nucleotides to the language of amino acids. And these are going to be our learning objectives. We want to be able to write the mRNA and write the peptide sequence given a DNA sequence, to talk about the process of translation, the different steps, as well as any post-translational modifications. As a reminder, here we are in our central dogma. We talked about DNA replication. We talked about transcription, going from DNA to RNA. Now let's talk about this final step, RNA to our polypeptide in translation. And keep in mind, in order for the protein to become functional, it's got to be correctly folded and assembled at the various levels of structure. So all of that you should remember from our earlier modules. Now, how does this occur? So how do we get from the language of nucleotides to the language of amino acids? Well, to do this, we use something called the genetic code. And the genetic code is said to be degenerate and universal. So every 3 nucleotides in our mRNA are read as a codon. And you can use this chart here to determine what that amino acid is. So if we start with our first position is a U, we would go to this line here, and then we would find the second position, say it's a C, we'd be in this box, and then no matter what the third amino acid is, we know, or third nucleotide is rather, we know the amino acid is serine. Now, this shows you clearly the degeneracy. So there are multiple codons that will give you an amino acid. Here there are 4 different ways you can get serine, 4 different ways to get that one amino acid. And this is true for almost all of them. The exceptions, tryptophan, there's only one way to get that. And then methionine, there's only one way to get that amino acid. And that's because it's our start. So every single peptide sequence you're going to make is going to start with methionine, meaning that every mRNA will start the reading frame, will start making proteins at the sequence AUG. We also see 3 stop codons, which is going to tell the ribosome it's time to stop making the peptide. Okay, so this is showing you how we get that from the DNA to the mRNA to the protein. So we start with our template strand, and we're going to use complementary base pairing to make the mRNA in the 5' to 3' direction from our template strand. Switching anytime we would normally put a T, we put a U. Remember, there are no T's in the language of RNA, we only use U's. And then we would break it into groups of 3, and we would use the codon sheet, which is this chart right here, to get the amino acids. And we build our peptide from N to C terminal. Okay, so there's a directionality here in the DNA, the directionality in the mRNA, and the directionality in the protein. And you need to be able to use the chart to do this sort of calculation, going from template strand to mRNA to protein. Keep in mind the template strand is different from the coding strand. Template is what is used, the coding is the other strand. And mutations that add or delete a nucleotide cause what we call a frame shift mutation. And this happens because it changes all of the codons after that change. So if we were to imagine the normal protein read, our big red dog bit the old man, if we add a nucleotide right here, now all of a sudden we've shifted everything. So our groups of 3 have shifted, and the language doesn't make any sense. Same thing if we deleted something or added, deleted, all of that are going to change everything after it. We call this shifting the reading frame. So we're shifting where we're making those groups of 3. And these are mutations that have really large and powerful effects. Okay, so what do we need to do translation? Well, we're going to need some ribosomes, some mRNA, some tRNA, protein factors. Remember, the ribosomes are made of rRNA as well as some protein. The ribosome has 2 subunits, a large subunit and a small subunit. And keep in mind it's got stereotypical weights for bacteria and for eukaryotes, for the large and small, as well as for the full, complete ribosome. Ribosomes also have a spot for mRNA to bind, and then they've got 3 sites in them, an arriving site, a P site, which has a very high affinity for transfer RNAs, which is always going to be filled. If you're filling a site, you're going to fill that P site first, and then an exit site, which has the lowest affinity, and it will shoot out the tRNA. And at one time, you can only have 2 of those 3 sites occupied with tRNA. So what are our tRNAs? Well, they're going to bring the amino acid to the ribosome. And each tRNA binds a specific amino acid, and it's going to have at the bottom here an anticodon, which is going to recognize the codon via complementary base pairing. When it has the amino acid attached to it, it's an amino acid tRNA, and that is added by an enzyme that's a synthetase. What I want you to know is anytime you see a synthetase here with the extra E in there, that means it makes something, but it uses ATP. So you don't need to know this whole process, but just know that it's the process by which we add an amino acid to our tRNA using the energy from ATP. Synthetase, making things with ATP, making things with energy. Now our mRNA is going to serve as the template for translation. So prokaryotes are polycistronic, meaning that there are multiple proteins made from one strand of mRNA, but eukaryotes are monocistronic, so one gene, one protein. And this is the stereotypical sequence here for our template. We've got untranslated region on both the 5' and the 3' end. We've got our start codon here, AUG, A being that +1 position, all the way to our stop codon, one of 3 sequences. The ribosome is going to bind at a stereotypical spot. Prokaryotes, we call it the Shine-Delgarno sequence, and eukaryotes use a similar sequence known as a Kozak sequence, which actually occurs kind of over the start codon. And in the middle is going to be our coding sequence. So we've got untranslated regions and our coding sequence. And the translation itself is going to have 3 steps: initiation, elongation, and termination. And we're going to focus on how prokaryotes do these processes. So for initiation, the initiation factors are going to use GTP with the mRNA to first bind to that small subunit before the large ribosomal subunit has even come into play. And it's going to use that Shine-Delgarno sequence of the mRNA to position it into the ribosome, again using complementary base pairing. It's going to help position it into the ribosome. Transcription, then the steps are going to follow. So once this is positioned, we can bring in our large subunit, we can hydrolyze our GTP, get energy from that, kick out our initiation factors, and now we can begin. So our first initiator tRNA holding the methionine starts in the P position. Remember, P has the highest affinity. And the next tRNA for our second codon is going to bind into the A site with help from a factor called EFTU that's been activated. So it has GTP bound to it. And that's going to bring in that second codon, that second transfer RNA. And then it's going to need to be re-energized. So it'll be recycled with the help of another factor, EFTS, which will allow it to get more GTP so it can grab another amino acid containing tRNA and go on from there. So this will continually be recycled using GTP as our energy source. Now the RNA, the ribozyme, so the ribosome itself can help form the bond between our amino acids, and it will be transferred onto that second tRNA. So we're going to break the bond between the tRNA and our first amino acid, and the growing chain is then going to be on that second transfer RNA, and that tRNA will then be kicked out of our E site. And that's going to occur with another factor, EFG, which uses GTP, and it's going to pull both the mRNA and the transfer RNAs down so that the transfer RNA is in our E site, the second one is in our P site, and now that A site is open. Because that tRNA is in the exit site, it can then be kicked out, and then we just have one tRNA in, and we have room now to bring in a second one. And this process will be repeated over and over and over again until we build our full chain. And then we will eventually get to our stop codon. And the stop codon doesn't have a tRNA of its own. Instead, it has a release factor that uses GTP to bind in when we get a stop codon, and it will break apart the whole complex. And the factors themselves, the ribosomal subunits, the different factors can be recycled, and our polypeptide chain is released, and now it can fold with the help of chaperones. Okay, so with that, we've reached the end of the material for the whole course, and we've finished the central dogma. So you are now ready for your final exam.